those can form polypeptide chains. Okay? Lipids also can form droplets, so they can form these more organized things that are sometimes called a mycel, which are basically just these droplets of lipids that will organize themselves so that they have their hydrophilic portions pointing out towards the water and their hydrophobic portions in the middle, very similar to a phospholipid bilayer. A liposome is one that is able to reproduce, so it can pinch off and make smaller droplets. It can incorporate more lipids into it, and some of them can also do simple metabolic reactions. And so these are things that are showing characteristics of life: the ability to do metabolism, the ability to reproduce. Again, remember, please, please, please do not be writing everything down word for word. Summarize. Put it in your own words, okay, but not everything word for word. Some liposomes were thought to help form the first what are called protocells. Okay, protocell is the first cell, like a prototype of a cell, basically. So since some of those liposomes can reproduce and do these simple metabolic reactions, like you can see here, taking glucose phosphate, okay, it's storing the glucose in the form of starch, okay, converting that to maltose, and releasing the phosphate, okay, that these were the basis of these initial protocells. Okay, so let's talk about the RNA World hypothesis for just a second. So the RNA World hypothesis is one that gives us a theory for the origin of genetic information. And so the big question here is, so what we currently have is DNA codes for RNA, which then codes for a protein. And then these proteins are some of the very enzymes that are needed for DNA to code for a protein, for RNA, and RNA to code for a protein. So the debate comes in is, how could this system have ever evolved? If the proteins that are needed for DNA, so it can be replicated, so it can be transcribed and translated, if you need those proteins to do that process, how could those have ever been present before DNA had coded for them? So the most commonly accepted solution for this is that RNA was the first genetic material, which that would allow for the enzymes to be made that could then help the DNA be replicated, transcribed, translated. So that RNA was the first genetic material, because RNA can serve as a template. It does it during replication. It does it during translation. RNA can also act as a catalyst. So RNA can self-replicate, which would support its ability to be the first genetic material. Okay. RNA can make a 3D molecule if there's base pairs occurring, depending upon the base pairing that is happening. RNA can act like an enzyme. Okay, so RNA can act as an enzyme. It can speed up peptide bond formation. It can act as spliceosomes to remove introns. Okay, and mutations can also occur in RNA as it replicates. So that means natural selection can act on the RNA so that the RNA that is the best at replicating and functioning would do the best. And so the thought process is here with this RNA world is that RNA is our first genetic material because it could do all these things. And eventually that RNA basically evolved into the DNA. And so protocells that may have had some of this and interacted in this world, that also goes along with our protobiont. Okay? Protocells and protobiont are used as synonyms. So in an RNA world, protocells that existed in an RNA world would have consisted of the lipid membrane, possibly a genome of DNA that had been transcribed into RNA, ribosomes that could make that RNA into a protein. Okay, so the lipid membrane pretty easily forms, especially if it's in water. The metabolic reactions that could have occurred in that iron sulfide hypothesis could have eventually made these products that are inside. You notice there's a lot of could-haves, maybes here. All of this is hypothesis and theory. Widely accepted hypothesis and theory, but still hypothesis and theory. So here we're just reviewing what we talked about a few minutes ago with that RNA world. That RNA is considered to be the first genetic material. It has the ability to self-replicate. It can act like an enzyme. In particular, an example would be what's called a ribozyme. A ribozyme is a piece of RNA that is catalytic, which means it can act like an enzyme. But they are different than an enzyme in the fact that they are consumed. Remember, enzymes are not consumed, they are reusable. And so this describes early Earth with this RNA, but no DNA and no proteins. So based on this information, we can form a basic timeline of the formation of the Earth and when life began. So if Earth formed approximately 4.6 billion years ago, the environment was too hostile for life until about 3.9 billion years ago. It took a long time for the Earth's environment to become hospitable to living organisms. And the earliest fossil evidence we have is from about 3.5 billion years ago. So if we put all that together and we try to narrow down when the origin of life is, hopefully y'all have already done that and realize that is somewhere between 3.9 and 3 billion years ago is our origin of life. So our first real life forms would have been prokaryotes. Remember, prokaryotes are bacteria, right? They're bacteria, that's a C. So they have no membrane-bound organelles, which would include a nucleus. Okay, and so the thought was, as these prokaryotes came about, DNA started taking over as the primary molecule of the genome from RNA, mainly because it is more stable than RNA and it is less likely to mutate. Okay? As the organism is going to continue to try to survive, it doesn't want its genetic material to mutate a lot every single time it replicates. And so with DNA being more stable, that became a better choice. So again, these first cells were prokaryotes. Okay, so again, bacteria, no membrane-bound organelles. You can see that here. There's the DNA. There's the DNA there in the nucleoid. They're going to have ribosomes. Remember, all cells have ribosomes because they need to make proteins. They're also going to have a cell membrane. Remember, you have to have a cell membrane to regulate what goes in and out of the cell. And you have to remember, too, that the atmosphere at the time here was anaerobic. So there was no oxygen in the atmosphere. And so that is telling us that for these organisms were unable to do aerobic cellular respiration because there was no oxygen present. So these organisms would have been doing just glycolysis. So you got to think back to your freshman year. Okay, remember, anaerobic respiration is glycolysis. Glycolysis is that glucose to pyruvate pathway that produces, you get a net gain of 2 ATP out of it. So they get a little bit of energy from it. And this also supports the theory of a common ancestor. Glycolysis is what is considered a conserved core process. Okay, and it adds credence again to the theory of a common ancestor. So our next pathway that would have come along after glycolysis would have been photosynthesis, would have been our next big biochemical pathway. Okay, photosynthesis originally, did not produce oxygen. Okay, so about 3 billion years ago, modern-day photosynthesis started taking place in the cells. And these cells resembled cyanobacteria, which is sometimes called blue-green algae. So they resembled cyanobacteria cells, and they evolved to start producing oxygen. And what this did is this started changing the atmosphere. So the atmosphere changed from what's called a reducing atmosphere to an oxidizing atmosphere. This atmospheric conversion laid the pathway for lots of other life forms to come about, to have this more oxygen-based atmosphere. So these cyanobacteria would form big, large mats, and they would collect together, and they would actually precipitate minerals out as sediments. These were fossilized. You can see that here in this picture here. Okay, here's the big mats of algae, that were doing this. And again, this was converting the atmosphere into an oxidizing atmosphere. So there were some consequences of oxygen starting to be produced and put into the atmosphere. No longer can you have basically life spontaneously arising from these non-living materials. Because remember, oxygen was very, very reactive. Okay? It has a very high, what's called an electronegativity. It pulls a lot of electrons towards it. And because it pulls a lot of electrons towards it, it makes it very reactive. And so that made these, that reduced the ability of life to form from these non-living materials. However, your organisms that can tolerate oxygen, they now have an advantage. And your obligate anaerobes, so these are your organisms that cannot have any oxygen present at all. Oxygen will actually kill these organisms. Okay, so your obligate anaerobe has to have an oxygen-free environment or it will die. They either did die, they either became extinct, or they found anaerobic environments. Remember, we have one of those kingdoms is those archaebacteria. And those archaebacteria would contain these bacteria that are obligate anaerobes. And so they are found in areas like deep mud where oxygen cannot penetrate, okay, but they are found in anaerobic environments. Some of the oxygen grouped together and formed ozone, which helped filter out the UV radiation, okay, which is extremely important for organisms because UV radiation can cause mutations. So now that we have a large number of these organisms surviving that are able to tolerate oxygen, our next step in the chemical pathway is going to be aerobic respiration. Aerobic respiration uses oxygen to make ATP. And remember, it is significantly more efficient than glycolysis. Glycolysis is only going to make those two, get you that net gain of 2 ATP. Remember, aerobic respiration gets you approximately 36 ATP. But again, that can only be done if oxygen is present to act as an electron acceptor. And we just mentioned that oxygen is very electronegative. So the electrons spend a lot of time and have a high affinity or attraction toward oxygen. So as these organisms are producing more oxygen and they're able to use their energy more efficiently, the evolution of the eukaryotic cell is going to start to come about. So right now we don't have any animal cells, we don't have any plant cells, because we need two things to have happened. We need compartmentalization to happen, form those membrane-bound organelles, and endosymbiosis to happen. Compartmentalization occurred due to the cell membrane folding inward. Okay, that's why they're called membrane-bound organelles. They're actually made of the same materials as the cell membrane, so they're made of phospholipids. And that cell membrane, says there's excess cell membrane. You can see it here, folds in to form the endoplasmic reticulum, the nuclear envelope around the nucleus, Golgi apparatus, lysosome, all those different compartments within the eukaryotic cell. And the benefit of these is it allows these metabolic pathways, these chemical reactions, to be isolated, to localize them, which is the easiest one I think to think about is lysosomes, right? Lysosomes digest food. And so if they're digesting something, we want to keep that separate. We don't want to break down the cell itself. Okay? And so that's one of the advantages of having these separate compartments. In endosymbiosis, we also talked about your freshman year in biology. So endosymbiosis is basically that theory that a smaller prokaryote was engulfed by a larger prokaryote, which is one of the ways that we led to some of the eukaryotic compartments, in particular mitochondria and chloroplast. So as these eukaryotes were coming about, we had the compartmentalization occurring, and then we also had symbiosis occurring between an aerobic heterotroph, so an aerobic heterotroph prokaryotic cell, and a eukaryotic cell. So you can see that's eventually what became the mitochondrion. You can see it here. So this originally, the mitochondria is thought to originally have been an aerobic heterotrophic, so that means it could use the oxygen that was in the atmosphere. It's producing that ATP. So an aerobic heterotrophic prokaryote that this larger developing eukarya took in. And eventually over time, the two were unable to live separately. And so when it went inside the original larger eukaryote cell, it was not destroyed, and their